all the event is end, please do not leave the Zoom meeting because maybe we will have some small talks in the breakout room. Thank you for coming today. Mas Faris, kita bisa mulai.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Honorable Dean of Faculty of Mathematics and Natural Sciences, Universitas Indonesia, Dr. Rokhmatuloh. Honorable Vice Dean for Education, Research and Student Affairs of FMIPA UI, Dr. Rika Tri Yunarti. Honorable Vice Dean for Resource, Ventura and Public Administration of FMIPA UI, Professor Yogi Yulizar. Honorable Manager of Research and Community Engagement, Professor Ivandini Tribidasari. Honorable Speakers, Professor Dr. Shamala Devi Sekaran and Professor Dr. Jacob Yan Mulyana. Honorable Lecturers, guests, ladies and gentlemen. First of all, let's say thanks to Allah Subhanahu wa Ta'ala who has given us guidance, happiness, healthy, and mercy so we can attend and participate in this special occasion without any obstacles. Good morning, ladies and gentlemen. Welcome to the 16th MIPATalk series with theme The Value of Science and the Role of Experts in Crisis Situations. Before we start, it will be better if we pray first. Pray according to each religion and belief. Begin. Finish. This morning, we have several agenda. Today's agenda begins with reading the rules for this event, then it opens with remarks from the Dean of Faculty of Mathematics and Natural Sciences, Dr. Rokhmatuloh, followed by a photo session. Dr. Rika Tri Yunarti will moderate today's event with the first presentation by Professor Dr. Jacob Yan Mulyana, followed by a question and answer session. And the second presentation by Professor Dr. Shamala Devi Sekaran, followed by a question and answer session, and ended with a closing. These are the rules and regulations during the event. During the presentation, the microphone from the participants will be deactivated by the committee. For Q&A, please use the raise hand or chat to Q&A topic 1 and 2 features on the platform. The participants are not allowed to use screen sharing and annotation features on the platform. MIPATalk can also be watched live through the FMIPA UI official YouTube channel. Ladies and gentlemen, let us welcome Dr. Rokhmatuloh, Dean of Faculty of Mathematics and Natural Sciences, Universitas Indonesia, to deliver his remarks.
Assalamualaikum warahmatullahi wabarakatuh. Good morning to our eminent speakers from reputable universities. Professor Dr. Shamala Devi Sekaran from UCSI University College Sedaya International Malaysia, as one of the Malaysia's first private universities. And the second, Professor Dr. Jacob Yan Mulyana from Tokyo Metropolitan University, Japan. And all distinguished guests, students, and colleagues, I bid you a very warm welcome to the MIPATalk Series 16, 2020, hosted by the Faculty of Mathematics and Natural Sciences, Universitas Indonesia. I'm very sure with the expertise and experiences from our today's speakers and moderator, we could gain valuable knowledge, and I truly wish we will have a fruitful discussion in the webinar. Starting at the beginning of the pandemic this year, MIPATalk series continues to facilitate discussion for all topics related to COVID-19. We are really hope that next year we are confident to continue hosting the MIPATalk series in online format with another interesting subject which will stimulate and nourish scientific gathering as well as to find solution for our daily life issues. We also hope that this webinar will encourage the international collaboration in research and education with our speakers from UCSI, Malaysia, and Tokyo Metropolitan University, Japan. And finally, with saying Bismillahirrahmanirrahim, I formally open this webinar. And thank you for your participation. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Dr. Rokhmatuloh, for the remarks. Ladies and gentlemen, now we will hold a photo session. You can turn on your video and please hold it for a moment. It will take a few seconds. Get ready for the first slide. 1, 2, 3. For the second slide, 1, 2, 3. Thank you. Ladies and gentlemen, I also invite Dr. Rika Tri Yunarti as a moderator. Dr. Rika, I hand it over to you to lead the session.
Okay, thank you very much. Good morning, everyone, and thank you all for finding time and visiting today's webinar. My name is Rika Tri Yunarti from Department of Chemistry, FMIPA UI, and today I will be the moderator for today's talk. And we also have this webinar through the YouTube channel of FMIPA UI, and I will also like to greet all the audience there as well. And on behalf of the FMIPA UI and all the committee, I hope we will have a very good time in this talk today. And our topic today is the immunity and the other side of COVID-19. And we will have two speakers today. First will be Dr. Jacob Jan Mulyana, and the second one will be Professor Shamala Devi Sekaran. And before we start, let me explain how you can talk with us during these talks. First, kindly mute your microphone during the session. And the second one, if you have any question during the presentation, just write them to chat in this Zoom chat, and then the committee will send the question to me. And then our agenda today will be the presentation from each speaker, and then after that we will have the question and answer session after each of the presentation. Now, for the first one, we can begin to our first speaker. Okay, yes, the CV, please. Okay, our first speaker will be Dr. Jacob Jan Mulyana. Okay, Dr. Jacob Jan Mulyana, or we can call him Pak Jan usually, or Dr. Jan. He graduated from the University of Melbourne in Australia in 2009, and then he used to work in Tokyo Metropolitan University, Japan, until this year, 2020. And then he's been coming to Indonesia like few times and came to our talks also, and already gave a lot of insight and contribution of our institution lately. And his research interest is for the renewable energy, solar water splitting, and material chemistry. He also already published some distinguished publication. In 2020, this year, the latest one is in Organica Chimica Acta 500. It's about the photo-induced water oxidation via cascade charge transfer on nanostructure bismuth vanadium oxide, TiO2 modified with dye and co-catalyst molecules. And for the presentation, Dr. Yan will have about 30 minutes, and after that will be followed with the discussion from 2 until 3 questions from the audience. And let's begin. Dr. Yan, time is yours.
Okay. Thank you very much, Ibu Rika. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody, I should say, because this is morning in Jakarta, although it's actually an afternoon in Melbourne. I'm actually in Melbourne, Australia now, and it's about 1:30 rather in Melbourne. I'm also thanking Dr. Rokhmatuloh, is the Dean of Faculty of Science in University of Indonesia, and also Professor Ivandini as well, who has invited me to give the talk today. Let me just share screen my presentation. How to Yeah, right. So Ibu Rika has already mentioned about my background, although maybe I have to just give a little bit of the more explanation, a little bit of my background. The last institution that I worked for was Tokyo Metropolitan University in Japan. And as Ibu Rika said, I'm working on renewable energy, solar water splitting, and also materials chemistry. So this is my research. My research is about renewable energy, how to convert water to clean fuel using hydrogen, using photo irradiation. Just to give a little bit of background, so we know that the Earth is hotter now and it's burning because of our continued use of fossil fuels. And my research tackles this climate issue by generating alternative clean fuel from water. These are the people that who have worked on my project. I had like a little space in the university that I worked last time. I'm not working there at the moment now. I'm not sure if I'm going back to Japan or not. First of all, there's Dr. Okta Suryani. She's got a PhD, a Doctor of Engineering in 2018. She's now a lecturer in MIPATalk. As Yuta Higashino is Japanese. He will continue his PhD in University of Sydney. There's also Indra. He's got the Doctor of Engineering in 2018. He's now a lecturer in Riau. And there's also Uji Pratomo, the Doctor of Engineering. He's got that in 2020 on February this year. He's now a lecturer in UNPAD in Bandung. All right, so I'm not going to talk about the renewable energy, although it's actually connected to this topic that I'm going to talk about. I'm going to talk about something that is related to COVID-19, although it's not exactly about COVID-19, but it's about the connection between the pandemic that we have experiencing at the moment. It's based on literature review. It's not based on my research, but it's actually based on a literature review. So I call this the other side of COVID-19 because there are many sides of this pandemic that we can actually study. All right. Let's have a look a little bit of the COVID-19 data as per 25th of October 2020. All right. In the USA, there are about 12,613,248 cases, with about 4 million recovered, to 260 deaths. In India, the second rank that we can see the COVID-19 pandemic. The third one is Brazil. So millions of COVID-19 cases. Some majority of them are recovered, but there are a lot of people who died because of this pandemic, right? Indonesia, it's really worrying. To me, it's quite worrying. In Australia, where I live now, we have about 27,000 cases, recovered about 25,907 deaths. As you can see from the statistics, you can see it from Johns Hopkins University website. You can have a look at that as well. What about the curves? We've been talking about these curves like many times in our social media, everything. Sometimes we have a lot of memes about these. The curves the one that we are so worried about, right? The USA, we can see there are quite a lot of first case, second hit. Maybe there'll be third hit again after that. We don't know, but it's still increasing up until now. This is the statistic from November. Indonesia is also worrying, particularly because it's still going, still increasing. I'm not sure about the health regulations and things happening in the country now, but China particularly has pretty much flattened. The curve is flattened, and it doesn't have any more cases, more cases in the country at the moment. The center one is quite worrying, of course, Indonesia now, but we'll see what happens, right? The COVID pandemic has created a global catastrophe, as we can see, and in last month, there was a joint statement by the UN bodies that the COVID-19 pandemic has led to a dramatic loss, of human life worldwide. The pandemic has created a lot of challenges to public health, food systems, and also the world of work. There are about tens of millions of people are at risk of falling into extreme poverty. Now there are about 690 million, undernourished people, which could increase up to about 132 million by the end of the year. So nearly half of the world's population now, the global workforce, are also at risk of losing their livelihoods because of this pandemic. So we know that this pandemic has created a lot of, problems, catastrophe, if you have to call it that, effect of the pandemic. Okay. So despite these catastrophic effects, have we seen any positive impact of the COVID-19 lockdowns on Earth? I like to say this. I'd like to ask this type of question to the audience because we're talking about Earth and we're talking about coronavirus on one side, but we also talk about the Earth on the other side. When we talk about the positive impact, we can talk about many things, whether it's actually from our internal situation or the things that we can reflect on, or something that we may have seen the impact, the positive impact. What I mean, we know that there are a lot of negative impacts, catastrophic effects, but have we seen anything that is positive from this COVID-19 lockdowns on Earth particularly? Right. So that's the question that I'd like to sort of ask the audience to sort of reflect on, think about. Maybe there's something that is positive. Have we seen that thing, or have we seen? Right. Okay, let me show you a lot of pictures around the globe during the lockdown period. This is Rome. It's very empty. There's particularly like exactly like, you know, there's nobody dares to sort of, you know, wander around the city in Rome on the day that the lockdown's happening. This is Paris. We can only see 3 people and quite huge number of birds flying around the Eiffel Tower. Right, it's also empty. Manhattan Street in New York during the lockdown, it's also empty. Usually that street is really busy on daytime especially, but during the lockdown, it's all empty. This picture might tell you something about, you know, the condition that we are experiencing, especially in the busy city like in Rome. This is the only way they can communicate with each other. You know, in the apartments and things like that, they communicate through the balcony. They sit down on the balcony. Sometimes they sing together. You know, sometimes they sort of wave the hands when a lot of health officer, you know, passing street and things like that to sort of save the patients. Yeah, so it's a lockdown mystery. So like it's everywhere. It's pretty much everywhere. It's a global phenomenon. All right, so this is New Delhi Gate. The top one was the picture taken on October the 17th, 2019. You can see that New Delhi last year, October 2019, it was hazy. It's got a lot of haze on it, on the city. People can't see the street because it's too hazy. On the 8th of April 2020, during the COVID-19 lockdowns, you can see on the bottom picture there, the air is clean, the view is also clear. Right, I'm just showing you this picture because I'd like to drive you to the situation where maybe we can find a positive impact of the lockdowns. Right. Right, so this is New Delhi. Hazy, clean, right? Hazy before the lockdowns and clean, clear. The air is clear, the view is clear during the lockdowns, right? Does it tell us something? It does tell us something, right? Now, I've been reading a lot of publications by the news, things like that, sort of, wonder how the virus might affect the people's life in a way that we might have clean earth because of the lockdowns. In Chemical and Science Engineering News, we can see that it says that the COVID-19 coronavirus quarantines cause rapid drop in air pollution and carbon dioxide fall rapidly as virus spreads. Coronavirus, wild animals enjoy freedom of quiet world. So I'd like to sort of, draw your attention. Are these the positive side of the COVID-19 or something that, we can think about? Yeah, the news, the publication and things like that, we can read from the media. Okay, so this is the satellite image of nitrous oxide levels in northern Italy during the lockdown. As we know from the news, from the TV, or from the newspaper, that the second country after China was actually Italy hit by the pandemic. And I'm sure that everyone knows about, how busy the hospital was in Rome, in Italy, and how stressful they are, the healthcare workers, to deal with the pandemic, and how it's really, distressed, creating the distressed situation to all people in Rome at that time. And Rome, Italy, or northern Italy particularly, began to experience the lockdown for all people in the northern part of Italy. At about, I think it was about February this year, 2020, until about March 2020. And this is the satellite image of the nitrous oxide level in northern Italy. And the nitrous oxide level began to decline on February 2020. As you can see here, the white spots is actually indicating the hotspots of the nitrous oxide level. And the red one actually indicates the lower levels of the nitrous oxide pollution around the area. So the NO2 level fell about 10% each week, as people in Italy left home less often. They don't go out from house that often. And the NO2 level had fallen by about total of about 50% on March 2020. For those who don't know about nitrous oxide is a byproduct of combustion emitted by cars, power, and industrial plants. It's not good for our health. The gas is harmful to human health and reacts to form unhealthy ozone. It's a different ozone type. The ozone in the ozone layer is probably different to the ozone. It's the same ozone, but it's actually different, leveling of ozone. So the ozone here is unhealthy ozone, and also it also creates the particulate matter, or PM, or aerosol. Right. So we know the danger of nitrous oxide level, and fortunately during the lockdown period, that level decreases significantly to about 50%. Right. Now we go to China. The same thing happens in China. We can see this is the satellite image by NASA, produced by NASA. In January 2020, the level of nitrous oxide is quite high, as you can see from the yellow sign here in about northern China, Wuhan, where the pandemic started. February, when the image was taken again in February, you can see it's clear. Actually, at this period, a lot of data scientists pretty much working on the area say that up to about 90% of the nitrous oxide drops in this area, because during the lockdown period. Yeah, so it's a similar thing as in Italy case, similar to China. Right. Okay. So I'd like to show you now how the pandemic, or the lockdown period particularly, has affected the daily carbon dioxide emission in 2009 in comparison with the 2019 emission. Right. So the top picture here shows us that there is a sharp decline in carbon dioxide emission in 2020 compared to that in 2019. You can see here there's a sharp decline as shown in the red graph. This is global, right? This doesn't represent any country, this is global. When we look at the second graph, globally, which is represented by the yellow line here, the most negative difference, this is the difference, the daily emission difference in 2020 compared to 2019. of the carbon dioxide emission. The most negative difference was noted on April 2020, when the majority of the cities are in lockdowns, right? And these graphs also are consistent with the COVID-19 curves in the US, in China, in the European Union, and also in the UK, right? So there is a link between the COVID-19 curves and the pandemic with the decrease in nitrous oxide and carbon dioxide emission on the Earth, right? I'd like to sort of draw your attention now to the comparison between the change of carbon dioxide emission with respect to the world event since 1900, right? There are a lot of declines in the history of our life since 1900 of the carbon dioxide emission due to a lot of reasons, you know, particularly the economic downturns and also World War. You can see that this decline is due to the World War II. We had a decrease of about minus 0.8 gigaton of carbon dioxide emission. Right, this area here where probably some of you were born in this particular year, we know there was a lot of the financial crisis around the year of 2000, early 2000. You can see suddenly the COVID pandemic has created or has caused an abrupt decrease of about minus 1.6 gigaton of carbon dioxide emission. And that is about 8.8% decrease, much higher than the one that was caused by World War II. And you know how long does it take with lockdowns? It's probably about 2 to 3 weeks, maybe about, in China, I thought it was about 2 to 3 months period, if I'm not wrong. I think that's really short, and it has created you know, a significant decline in carbon dioxide emission. You can imagine, right? Okay, now which sectors contributes to the carbon dioxide decline? This data shows you about when people are locked down, when people cannot go out, when they have to stay at home in order to flatten the curve, right? Now, the sector that contributes most to the carbon dioxide decline is actually the ground transport, where because we, you know, travel around, you know, from house to work or something like that, we have that limitation because of the pandemic. So the ground transportation contributes to about 40% of the decline, followed by power sector. Power, I think power, because we need power, even though we stay at home, but we need power. Industrial sector, and the aviation sector, we know that we don't have much privilege to, you know, fly everywhere, you know, because of this pandemic. International shipping also contributes about 66% of the decline. Residential, public, commercial buildings contributes to about 3% of the carbon dioxide decline. Right. Somehow, there are a lot of researchers who found the opposite finding, who presented the opposite finding. And this comes again from China. In China, the studies shows that up to about 90% reduction in emissions. That's the data that they got from you know, the measurements. Although the 90% of the reduction in emissions, they still see that certain areas in China remains hazy. and certain areas experience aerosol and ozone enhancements as well in certain areas, despite the 90% reduction in emissions because of the COVID-19 lockdowns. Now, this is somehow opposite to the other finding that the other people presented. But this research also say that because, even though the 90% reduction in emissions, and it still creates haze and ozone and aerosol pollution. That happens because of, at that time during the lockdowns, there was a high humidity condition in China at that time. And the fact that the chemistry processes associated with the aerosol and haze formation is a complex process. It doesn't represent, the COVID-19, the reduction because of the COVID-19 data may or may not represent the impact to the aerosol and haze formation. That's what they say. And the nonlinear ozone and aerosol chemistry also contributes to the discrepancy between the data presented by other people and the data presented by this journal. Right. Now I'd like to show you as well how the wildlife suddenly enjoy their life in the city during the lockdowns. Santiago in Chile, suddenly they see the koga spotted jumping onto the wall in the city. This is like, wildlife. They haven't seen that wildlife in the city for maybe, I don't know, 100 years, I'm not sure. The wild boars in the street of Haifa in Israel or in Palestine. The dolphins, which are not usually coming up in Bosphorus in Turkey, suddenly they appeared like, in the Bosphorus in Turkey. Right. So maybe about, I don't know how many weeks, maximum probably about 3 to 4 weeks, the wildlife enjoy their city life during the lockdowns, and suddenly now they have to go again, I don't know, somewhere on the earth because the human activities begin to be, starting again because of the relaxation, because of the easing of the pandemic in some areas. Right. I'm not sure how long I think I still have a lot of time anyway, but I'm not going to talk that much again because this is the one that I'd like to show everybody how a catastrophe actually leads to healthier Earth. I'll repeat again, how a catastrophe can lead to healthier planet Earth. Now we have a lesson from Chernobyl. I'm sure everybody knows Chernobyl, where the nuclear reaction explosion occurred. I forgot what year that was, probably in about 1990s or something. And it was published in Netflix and narrated by David Attenborough. We're not going to watch the whole movie, the whole YouTube, but I have cut some part of the YouTube so that we can enjoy viewing that. So I need to stop share screen of this, and I'm going to share screen of the YouTube channel. The admin, could you tell me if something doesn't work out? So I'm going to play now. Okay, so that's the presentation. I have to go back to my slide. Right, just watch something that I hope tells you that a catastrophe can lead to the healthier planet Earth. And this is the end slide. I hope that everyone has an answer now, at least the thinking inside our heads. To save our planet, should we wait until a pandemic or a catastrophe occurs? Thank you very much.
Yes, thank you very much, Dr. Jacobian Maulana. Yes, it's something that we can, how can I say, see the effect of this pandemic. Actually, there are two effects here, the positive effect and the negative effect, of course, the biggest one. And to discuss more about this, we will have some questions, Dr. Yan. And Dr. Yan, we already have, let me check, 2 questions already here, and then I will read it for you. And the first one is from Hanzola Guzman. And yeah, I would like to ask for Professor Dr. Jacobian Maulana, few countries apply herd immunity method to overcome the pandemic, such as Sweden. What do you think about this method compared to lockdown, which used by almost countries in the world? What the scientists' prediction when the pandemic will be over or finished? Okay.
It's a very tough question. It's a very tough question. It's outside my field of research and it's outside my thing, but I try to answer that. The herd immunity, this is actually the one that I got from my wife. My wife is a doctor. she told me about many things about herd immunity. It has a lot of, it takes time, and it might have a lot of victims as well as well, I have to call that victims because people can die because of that, and then eventually we will build the herd immunity. I think Professor Sekaran will also have an answer to this one as well. To me, to my experience, lockdowns is pretty effective in terms of flattening the curve. To my experience, it's at least in Australia, right, where I live. I've been in lockdown for about total lockdown for about 3 weeks, I think, in Australia, in Melbourne, particularly because Melbourne, Australia is the city where the virus takes more effect than other cities. And now, like, we've been having the lockdowns, total lockdowns for about 3 weeks, and now we don't have any more cases, like 0 cases for about at least 20 days now since the last lockdown. So it's quite effective, and that's within 3 weeks. I'm not sure how long it takes for the herd immunity to actually take place. I'm not sure about that. Maybe I'm not the person who's the best person to ask for that. From my experience, I think lockdown, at least from my experience, it's quite effective. And what's the other question? Sorry.
Okay, what the scientists' prediction when the pandemic will be over or finished?
There is no a definite time when this pandemic will end. It depends. There are a lot of politics, associated with the vaccine availability and how that vaccine can be effective or non-effective. It's kind of multi problems, if I can say that, and it's no one can predict when this is gonna end. Maybe this year or next year, maybe the week after. We don't know. So we don't really know about that. As I said, maybe I'm sure that Professor Sekaran will also have the answer to this as well.
Yes. So I think it's also not because it. It not only about the health problem anymore, but it includes so many aspects.
Exactly. So many aspects. It's really,
That's right. And here I have a second question for you. From Arvin Fardiansa, there is no doubt that COVID-19 has brought positive effect to the environments, mainly in the reduced CO2 and greenhouse gas emission worldwide. However, as the world healed, the human activities also tend to increase, which lead to the increasing number of greenhouse gas emission as well. Thus, is there an efficient way to significantly reduce the emission, but not immensely affecting the human productivity?
Again, this doesn't attract a simple answer. This is also a complicated answer. Climate change is also a political issue. It's real, although there are a lot of climate denials. Yeah, but it's real. It's facing us. It's the earth is burning. We can do many things. The simple thing, maybe, I don't know. It's hard for you. I know it's hard for you, but can we use the gadgets wisely? Right? If we can use the gadgets wisely, the mobile phones, Instagram, Facebook, whatever, we consume a lot of data. Consequently, we use a lot of energy. If we use a lot of energy, then the carbon dioxide emission will also increase. Consequently, there are a lot of wise things that we can do at home, personally, and things like that. And in terms of the workforce or something that can lead the industry forward without creating a lot of burden on the environment. My research is concerned with replacing fossil fuels with something cleaner. That's the thing that we can also develop. In Japan, there are a lot of industry now. Cars replaced with the non-fossil fuel cars, such as hydrogen energy or battery. In terms of when we think about something produced by the USA, Tesla, for example, the cars, it's all the vehicles that don't rely on the fossil fuels. Fossil fuels is actually the main culprit for this. If we burn many fossil fuels, then the earth is gonna warm and hot and hot. Yeah.
So still the main focus is to reduce the using of fossil fuels.
To reduce the use of fossil fuels. That's our task.
Yes, that's right.
But as I said, we do not have to wait until a catastrophe happens.
Yes, that's right.
Right.
Yes.
We should wait, and we should, of course, this pandemic has created a lot of mystery, catastrophe, and things, but there is a positive side on that one, as I said. I'm not saying that the pandemic actually creates, it's good for us. No, it's not. But in fact, it makes the earth healthier.
Okay.
That's the thing. Think about.
Yes, that's the fact.
I'm not pushing you to think about that, but something that's we should think about.
Yes, that's the fact that we have now, right? Yeah, and then beside all those negative things that we get from the pandemic, there is another thing that came up actually that it didn't design earlier, but it's like that, and then so it's very interesting actually, and from the video that you showed about the Chernobyl that I saw also that documentary, I also saw the series itself with some drama there. It's very interesting actually, but it's really sad actually. Yeah, but still we can see a lot of aspect over there that created after the what do we call it? We call this pandemic, and that the Chernobyl we call it, how can we say, disaster or something?
Disaster.
Like that. Yeah. So, but still, we are hoping that the world should going to be better soon. Yeah, I hope maybe in this month or next month.
Hopefully, we all pray for. We all pray for it.
Yeah, and we also can start again to produce more renewable energy. After this pandemic. Yes. So thank you very much, Dr. Yan, for the a very interesting presentation. And then I hope we will have another session with you, of course.
Sure. I'll be online, but probably not that active because I have to pick up my child.
Yes, sure.
So I'm sorry I have to leave for the moment, but I'll try to enjoy it again.
Okay, then.
It's great to see you, and I'm thanking everyone for.
Yes, thank you very much. And I would like to summarize a bit about this talk from you, that COVID-19 pandemic has created a global catastrophe with dramatic loss of human life worldwide, challenged public health, food system, and the world of work. But on the other side, COVID-19 also affected the air quality in the atmosphere that has caused an abrupt decrease of CO2 emission. And then that will affect the plants and the animal life over there also in our world. Okay, thank you so much, Dr. Yan. Yes, and be careful.
Thank you very much.
Thank you very much. Thank you.
Yeah.
Thanks to all. Professor Sekaran, I'm sorry. Thank you very much.
Welcome.
See you later.
See you later. I'm still online anyway.
Okay, take care. Bye, Yan.
Yeah.
Thank you so much.
Bye.
And then now we are going to the second session with the second speaker from Professor Shamala Devi Sekaran. Okay, I will read a brief CV of Professor Dr. Shamala Devi Sekaran from UCSI University. And she graduated from the Royal College of Pathologists in UK, and now she's a professor in Faculty of Applied Science, UCSI. This year, professor, actually, right?
Yes.
You just moved. Yes, with the research interest of viral pathogenesis, diagnosis, and antivirals. So I think this second talks also will be very interesting because I bet you are really intact with this our current situation for the research. And okay, Professor Shamala, time is yours. You have about 30 minutes. Thank you.
Okay. I will just share screen.
Yes.
Right. Okay. Yeah.
Okay.
Okay. So, let me put it on show. Okay. So let me just begin to first qualify myself. Yes, I am a virologist, and I basically do quite a bit of diagnostics work as well. And with the COVID, I have been involved with the diagnostic laboratories. As you know, the FRC pass is just a qualification with with UK to enable me to perform diagnostics for patients, basically. But basically, I'm a scientist who's been working on dengue for many, many years. But maybe as once the lockdowns and once things are better and the grants are available, I might decide to go and work on COVID-19 as well. Okay, so let me just start first of all with what are the questions we basically need to address. Probably to address those questions on immunity, we need to understand the relationship or interactions between the virus and the host's immune responses. To do that, we also need to understand the pathogenesis of the infection. That means, how does it cause the damage that it causes, all right? And this knowledge is especially useful if we want to identify therapeutic tools, all right? Which state of the infection we want to control and how do we repair it, okay? In this situation as we are in now, we do know that there are individuals who are asymptomatic, and in Malaysia we see close to 70% of people who are asymptomatic. They are infected, but they don't display any signs of symptoms. Okay. At the same time, we also see a fair number of people who end up with very severe disease, and we're wondering what the virus has done to evade the immune system as such. Okay, So among those questions that we probably would like to answer is, are you protected once you are infected? And how long does that protection last? Does my age have to play a role? Okay. Will the type of infection I have, whether it's mild or asymptomatic or severe, affect how my body remembers it, and will it protect me in the future? All right. So then there are a few others, and hopefully I'd be able to come to it too as well. All right. I think these questions are essential, where we want to consider whether we are going to be vaccinated or not. But to start with, of course, I think it's good to note that this is what basically diagnostics are all about, right? So you have this incubation period in the beginning where you are asymptomatic. Some of us then display fever, all right, after about 7 to 14 days. And it is these people who get the mild symptoms, and then quite a number go on to recover. It's only a very small percentage basically that end up with severe disease. So you have an asymptomatic and a symptomatic phase. During this time, people have noted that you are actually shedding the virus, and this shedding period can go up to 4 to 5 weeks at least, and you still shed the virus. At the same time, after 7 days of clinical disease, you begin to display, your immune system has begun, and you see levels of antibodies. But we also see these antibodies in patients. So knowing this curve then enables us how to develop diagnostics, okay? So currently it is PCR. You also do have the antigen detection test, and you have the different antibody assays, okay? There are issues with each of them, so how to use them is important, but they all can be used. What's important is any assay that you use, you need to know how to interpret them, all right? And for COVID-19 right now, we actually advocate using at least 2 tests, different sampling times, okay? So there is a diagnostic workflow, but I won't go into that as I want to just go into immunity per se. Of course, in line with our immune response, we forget about cells because, you know, being a virus, it enters the cells. So it is our cellular immune response which also plays a very important role. But these cells are more difficult to diagnose, and hence many people just leave them out. But we will address them. Okay. So let's start with just some background first. We know that our immune response to viruses are very heterogeneous, right? You have an early phase, all right? In the initial phase, when you first get infected, it's largely cellular, and it largely involves a lot of soluble factors as well, right? So against a primary infection meaning implying this is the first time you're being infected. So what the body does, it creates an antiviral effect. What that means is the cell that is infected will release these soluble factors that will protect the surrounding cells from infection. So that's why you call it antiviral effect, all right? But the innate immune response is also important because it is this system that will seed ours, the other part of the body, the other part, potentially the adaptive arm, which is your specific arm, right? And the specific arm is basically made up, as we all know, of cells, largely your cytotoxic T cells and your helper cells, and antibodies as well, right? But it is the cells that come up first before the antibodies. So therefore, an antibody basically does not contribute to recovery in a primary infection, but they do have a large role to play when you get subsequently infected, okay? Hence, that's why they measure mainly antibodies when they are developing vaccines, okay? So what has been shown for COVID-19, okay? So before that, this is just a brief overview of immune responses because maybe there are a lot of lay people around. So this side on the left, you see your innate immune response, which is rapid, immediate, it's non-discriminatory, and it occurs almost instantly. And it's made up a lot of cells, but it's also made up of a lot of factors once these cells are activated. Okay, these innate arm is the one that will then seed the adaptive arm, which takes time. maybe 3 days, maybe 7 days, sometimes 14 days or weeks even, okay? And there you have two aspects of it. You have the humoral arm, which is basically just little Y-shaped structures, which we call antibodies. And you have the cellular arm. For any viral infection, a cellular arm is important because cells that are infected, your antibodies cannot enter. You need a cell to kill a cell. However, in this instance, this is what we call a cytopathic virus. So what happens, what it means is that when it infects a cell, to get out, it eventually will lyse the cell. So they come out of the cell. Then antibodies do have a role. Antibodies are basically found free in circulation. Okay, so that's just basically a brief overview of it, right? So let's look at currently what we know, especially, of course, we have to learn from other coronaviruses like your SARS and MERS, and also the common cold, all right? We do know that, okay, there are antibodies, they do persist, and they can actually protect you, all right? However, at this point, the immunological and biological data has not been properly done, so we do not know precisely how long the protection will be. Now, our immune system basically has many ways, all right, by which they will keep off the viruses. First of all, we need to have these B cells. They will produce antibodies, as I said. And then if these antibodies disappear, we do have a backup plan. All right, because what happens is these B cells become memory cells, all right? And when the virus does enter our body again, it is these memory cells which are important that get reactivated, all right? In COVID-19, of course, it's too early for us to talk about memory B cells, but maybe in a subpopulation, we could, all right? But the data is still incomplete, all right? The B cell memories are also difficult to locate, all right? We do know that, okay, memory B cells or long-lived plasma cells can be found in the bone marrow, right? But so far, we do know that they do exist because there are people who appear to be getting infected again, all right? So to look at other coronaviruses, for SARS and MERS, yes, it has been persisting, all right? But it does wane and it does not drop off. So that means the memory exists, okay? We do not have enough data as such, okay? So I think I have done this slide, so I'm just going to go through it. I forgot to delete that one, okay? Right. Now, our immunity, apart from what I've just told you, does not rely on antibodies alone. We do have cells, and for viruses, these cells are important. When the virus is in a cell, antibodies cannot get in. So you need these T cells, right? They will patrol the body. They will then try to destroy these cells. And these T cells have got a long lifespan. They are for about 20 years, they are still around, okay, compared to your B cells and your antibodies, all right? But they're more complicated to probe, all right, than antibodies so far. But they do exist, and I think studies have been published to show that T cells do exist, all right? And one recent survey actually showed that there are T cells, okay? There are T cells that recognize the coronavirus in all of them. So most people appear to have a very good T cell response. And I think probably this is the response you would see in the asymptomatics, because it is the first one that comes up before your antibodies are produced, okay? Okay, I just ignore this picture. This is just to tell you all the different reactions that T cells can do. So if you were to have an antigen or if you're vaccinated, this antigen has to be taken and shown to your T cell, which will then get activated. Maybe it'll help the B cells produce antibodies, or it'll activate by releasing a host of soluble factors, which we call cytokines, by the way, and that then goes on to perform their function. Okay. But what we also do note is that among T cells from the different coronavirus groups, there is cross-reactivity. So they have reported that with SARS-CoV-2 in people who have not been exposed to the virus, all right, maybe they've had a common cold, so which means there is cross-immunity among the coronaviruses. So hopefully, I don't know, I would think that in this day when they're trying to develop vaccines, that you would probably want to find the conserved region among these human coronaviruses and use that in the vaccine. However, this cross-reactivity we also must try to remember. Is this in part responsible for the differences we see in the severity and the symptoms between individuals? So could the cross-reactive T cells cause more harm than protect? All right, so being cross-reactive does not always mean it's a good thing, as we know in dengue, all right? Yeah. So this is actually proposed by a researcher, mainly the Enrico Maggi actually, on what the host immune response to SARS virus is like, okay? So you have entry, this aerosol uptake, all right, by AC2 expressing target cells, which we will show you later where you find these, okay? And what happens is the virus is appearing to dampen the antiviral response. And these type 1 interferon responses are dampened. The antiviral effect is not created. And because of that, we get uncontrolled viral replication. So what happens is, as a result of that, you get increase of cytokines being produced, and that will basically start to bring in all your PBMCs, neutrophils, monocytes, and they will then start to also get activated in the process of trying to clear the virus. They will release cytokines, and that's how the cytokine storm began. Now, in the lung, when you have all these factors, they can actually cause damage, get more cells infected, get more cells lysate, and ends up with lung immunopathology. Apart from it, you also get T cells, which will then emerge, and they will also further exacerbate or make the response worse by releasing more cytokines. Okay? So, in the process, okay, is at this time, as you see, cells are the first to respond. You get antibody production as well. And we think that the first type is always neutralizing. So B cells are activated and they start producing neutralizing antibodies. Okay? But this, however, is still speculative because different people have done small parts here and small parts there, and we're trying to put them all together to form a picture. Right? So it still has to be further validated as such. Huh? So this is where I wanted to show you where you find these receptors. Actually, these red areas are red areas that have the highest concentration of the receptor for the SARS-CoV-2. It's not in the lung, it's in the intestines, in the kidneys, and in the testes, right? But it is moderately level in the lungs, but a little higher in the heart. So when patients with patients have heart failure, they increase the level of AC2 receptors. Okay, so therefore you find that, nevertheless, we find because of the exposure is aerosol, the lungs are the ones that probably will be the first exposed. And there, of course, it depends on your lifestyle, on your severity of lung disease you have or not have, if you're a smoker, if you have any other underlying conditions, then this expression of ACE2 will be higher. So this, then you can think about the type of people who are getting sick are really in the upper elderly age group, where they really then end up having to be on ventilators. But not to say that there are not in the other groups. Therefore, in the other groups, there probably is an underlying factor that has increased the level of ACE2 in the lungs. Okay, and therefore you get that. Okay, so in the innate arm, okay, this is the first exposure to the virus, first of all. We do have an effective innate response, but it's not working. Why? Right? We have that IF interferon gamma, which will control it and then see the adaptive arm, but it's not. What has happened is the virus has found ways to antagonize it. All right? It evades by being recognized by our innate receptors that we have, which we call PRR. These are pathogen recognition receptors. So these are receptors we have to recognize that there's a pathogen that has entered. What they do is they antagonize the action. So downstream events that occur so that interferon, type 1 interferon can be released are not activated. So it stops the signaling there. Okay, you need the release of type 1 to go and tell the cells around, okay, what they have to do to protect themselves from this virus that's going to come once this infected cell has lysed. Okay, so this failure to elicit this initial type 1 interferon is what correlates with disease severity, because you get uncontrolled viral replication. And it's this people with high viral load that are the transmitters at the moment. Okay. Okay. So as also another point to consider, that what has been shown for some people is your myeloid cells. Now, myeloid cells basically are your neutrophils, your basophils, your macrophages that comes out from the myeloid area. Okay, and they are actually, I've actually been told that they are dysregulated, okay? And the responses that potentially drive the syndromes that you would see in COVID-19, which is a respiratory distress syndrome, acute respiratory distress syndrome, the cytokine release, and lymphopenia, where these cell counts are become very low, okay? So this, what I'm going to show, is basically knowledge from other coronavirus, because it's not completely studied, but has been suggested, all right? So what they note is that your alveolar macrophages in the lungs change the phenotype, and instead of becoming one that is supposed to get rid of the virus, it becomes pro-inflammatory and starts to relieve your massive cytokines that you get. All right, you don't get the interferon signaling, but you get the increased cytokines, and those cytokines will start leakage to occur. Now, this, of course, will also, in the presence of antibodies, will actually exacerbate the whole reaction, causing more injury. So, and this whole process, of course, recruits a whole lot of cytokines, as you would see most papers will tell you, and the favorite word they have is the cytokine storm, right? In the pulmonary epithelia, in the lung area, further amplifies the inflammatory reaction. Okay, so you get complement activation, you get a delayed interferon response. There's another issue which I will come to in a short while, and that is how antibody actually enhances viral replication, or what we call antibody-dependent enhancement. Okay, so moving on, okay, from the humoral, we come to the cells. Now, we do know cells are very important. They help B cells make antibody. They also are cytotoxic. If a cell is infected, their job is to get rid of it, okay? So, but this whole cascade in COVID-19 has been suggested that it's been dysregulated. So, two questions we ask. What contribution of T cells in controlling the virus and for causing tissue damage? And do they contribute to protection? All right. What have they seen clinically is decreasing numbers of T cells because they're destroyed, and the extent of the decrease is said to correlate with disease severity and mortality. Okay? They also see an increase in these cytokines, a pro-inflammatory group, which will inhibit T cell circulation. So instead of allowing the T cells to move through, they are retained at the lymphoid organs. And the antiviral response is there. They note that there appears to be a good antiviral response. There are loads of CD8 T cells, and most of the time, those T cells are recognizing viral epitopes. But how are they functioning, and what is their response is still, I think, an area of research that they are still doing. But generally, they think there is a dysregulation. And this regulation could be at the point of regulation of the immune response itself. Okay? So by and large, the T cells are activated. But what is noted is they are exhausted. Okay? They exhibit a trend towards what we call T cell exhaustion. Okay? The cytotoxic sex, which they're supposed to do, is decreased. There's loads of antibodies. You see a lot of Th2 cytokines, and these cytokines will promote antibody production. Okay. So, but what is needed is a Th1 response, which is a response that results in a CD8 positive T cells and results in a cytotoxic T cell response. And this is where it needs to be properly controlled, not to cause lung pathology as well. So this balance between the two types of cytokines, Th1 versus T2, Th2 cells, and the cytokines they produce are important. Yeah? But and we are still at early stages. I'm trying to look at the literature to see, and sometimes it's kind of confusing. So I'll just give you what I am able to compare cellularly between these three viruses, which is the two previous SARS and MERS, and currently the SARS-CoV-2. Okay, so this we know very well already. They basically have a good response, okay, and basically you have good T cells, you've got good antibodies, right? But the virus basically died down too fast, and so now we don't have enough, we could say, patience or enough data to further extend to it. And the same thing happened with MERS, but we did that. But this was clearly seen. Okay, so this word, if you want, CD881 is actually part of the molecule present on T cells that actually involved in forming the synapse. Okay, the synapse is between the T cell and the antigen-presenting cell, right? And they are actually early markers that people look for. Huh? In SARS-CoV-2, okay, we don't know what is the repertoire or the type or the strength of the T cell response yet. They do see that NK cell circulation is reduced, and you do see that the different T cell subsets are reduced, hence lymphopenia that you see in patients. Okay, so this data they are still collecting. All right, hopefully we will know something about that. Oops. Now, with regard to antibodies, okay, this antibodies is much easier to measure, so you have a bit more data. Right, with SARS, they seroconvert by day 14, and they have these antibodies that are neutralizing for up to 2 years. And they are all in the receptor binding domain, mainly. Okay, of course, in other domains you have. So, but what they note is that patients with severe outcome, level of antibodies are delayed or weak. Right? This is also seen with regard to MERS. The seroconvert, but in those with severe outcome, again, is delayed and is weak. Their antibody levels are lower. Now, in SARS-CoV-2 currently, antibodies come up after 7 days, okay? There is seroconversion in most patients that have recovered, right? There's also seroconversion in people who have died, because they do measure that. But what they note is that most of the time, the specificity for the receptor binding domain is infrequent. But what we also note is that they do not have a weak antibody response. In fact, they have a higher antibody response, a very good antibody response. So the question to ask is, what role is that antibody playing? Which we don't know. Okay. So now, this whole group of viruses, I look at SARS, MERS, and SARS-CoV-2, they are beta coronaviruses, and they are well adapted to evade our immune system, all right? And they're very good at dampening it, all right? But they have a pretty long incubation period. Okay, it can be anything between 2 to 11 days, which therefore tells you that they have ways and means to avoid being detected or evading the immune response, okay? So one of the things, of course, the first thing as most viruses would do is to, the immunocompromised are the first ones to be targeted, okay? So we have, as we have seen in the earlier slides, early inhibition of your type 1 interferons. We have inhibition or actually decrease of your innate response in terms of the lymphopenia you see, so macrophages, DCs, and NK cells, they basically die, okay? But you also see an inhibition of the cellular response, mainly the T cells, okay? Not so much the antibodies, okay? Now, it is also noted that high viral loads overcome the barriers of the immune response. So that is probably a strategy. Initially, by dampening the type 1, you enhance viral replication or you aid multiplication of the virus. In that way, you can dampen it, okay? So I will skip this slide because it's just a repetition, okay? Just the next 2 slides to show you some examples of some studies that they have done, okay? if you look at the slide on the right, between the colors, you would see that the high levels of antibodies are noted in the critical cases, the green line. The critical cases have got pretty high levels of antibody as compared to those with mild disease. You know, the mild disease are the blue ones. Okay? If you measure the antibodies, there appears to be quite a high level, but IgM always will come up before IgG. If you look on the side to the left, the levels show that between the critical and the non-critical cases, they have equivalent amounts of antibodies. So the non-critical could also be those that are asymptomatic, right? But yet, in a proportion of people, that same level of antibody is not a helping. So this is a study done on 173 patients. So by 11 to 14 days, about 80% seroconverted. There were some that did not seroconvert early enough. The first 7 days, the antibodies were not present, all right, in quite a majority. But by 2 weeks, most of them seroconverted. But what is also to note that the presence of virus is still around, okay, despite you having that level of antibodies. Okay? So the second study is even the second part of that study, they actually took the severe cases. This is 9 cases of disease severity. And just to note, all right, for you to know how to look at this is basically one, you would note that the green line is the antibodies. The purple line in all of them is the virus. The other lines are all antibody lines. The purple lines are all very high, despite the presence of antibodies in all of them, and pretty strong antibody response. It's not weak. Totally together, you will see that they are high. So there is a rise in antibody, okay, as we see here, not always accompanied by viral clearance. So which implies that antibodies are not sufficient to clear the virus. And they remain elevated for almost 2 weeks after that, all right, 2 to 3 weeks. So what you also note is the critically ill patients have higher antibodies than the non-critical cases 2 weeks after getting sick, after onset of disease. Okay? So is there a strong positive correlation between severity of disease and the antibody titer? It's a big question, all right, that needs to be addressed. So this is another study, but maybe I will not go through it. But to look at this one at the left-hand side, they measured IgA and IgG as well, and IgA actually appears much earlier, okay? Again, here you need the small dotted line is your severe cases. They have so much higher levels of antibodies, be it IgG or IgA, yet they are not doing anything. Right? They are not protected. This is in severe disease, severe cases only. So during the first week of COVID-19, the sensitivity of the antibody response is low. All right. So what do the above results basically tell you? First thing, a high titer of total antibodies may be considered. Is this a risk factor for critical illness? Okay, is this independent of older age, of being male gender and comorbidities? We don't know yet, all right? We don't know yet, all right? I say older age because we know elderly, we are slightly immunocompromised. Males because of the higher incidence of ACE2 receptors in the testicular cells. And of course, people who are diabetic and obese. Generally, most diseases, when you are diabetic or obese, you are compromised. So now, if you see these things about the antibody, can we use the high antibody titer as a surrogate marker for disease severity? Maybe you would then progress to a more severe disease. Okay. This might also indicate that is the antibody doing like what it does for dengue, right? That means there is an enhancement of viral infection. That basically the antibody binds to the virus and then enables entry into cells that contain Fc receptors where the bond breaks and the virus replicates. So which implies these antibodies are not best antibodies to have. They're not neutralizing, all right? Maybe they are enhancing. or the bond is weak, therefore upon entering the cell with the change in the pH, the bond breaks, the virus is free to replicate. Okay? So that then comes me back to, I think, a question that was asked in the first session about, general immunity in the population. So many are trying to predict, all right, once a high enough number of people are infected and they have become immune, then the pandemic will subside. And that's basically the concept of herd immunity. That is the percentage of people who are immune. Generally, you want to reach 95%. Then you know you have reached herd immunity. And therefore, the virus will struggle to find susceptible people, right? There'll be no more susceptibles, okay? But what about the fact that each time there are different versions of the virus that comes about and it starts to circulate? So then we question using herd immunity as an approach to controlling the pandemic, okay? With these different versions, the antibodies that you get here, does it protect, right? Does the immunity you get initially protect you to a different version? So right now, of course, we're seeing sufficient evidence. We are still in early days. Even though we're seeing we've got so much patient material, so much data, we still don't have proper consolidation, right? Okay? To determine what levels of antibody to which target we should have and to determine what immunity is. And sometimes when you think you have been infected and you think you are safe, so you have a positive result, you've got so much antibodies, then you ignore the public health advice. Then you start another spike. Okay. So what is missing from all of this? Firstly, information from asymptomatics, which have not been obtained. And I know that 70% are asymptomatic. We should really start looking at these people. All right. These are the people producing the right kind of immune response. Right. Information about the immune profile, it will probably come out soon. Regulatory mechanisms. Okay, virus escape mechanisms will be ongoing as long as there are scientists who are, have a passion for it, you'll find. But what's important to know is to look at what is dampening the immune response. What is that doing at the early stages to dampen the type 1 interferons? And finally, the last part of it is that we need to know what epitopes to use so that we get this sterilizing humoral and cellular immunity. Sterilizing so that that's it, you are protected, right? Nothing will shake you after that, right? But what each part of the virus should be directed to? And there are more unknowns. All right, what percentage becomes immune if the virus is what percent of people become immune to the virus if exposed? There are always a percentage of people who are non-responders, and they will be the ones who maintain the virus, okay, in the population. How good is this immunity? Yeah.
Sorry, maybe 1 more minute.
Yeah, I'm just. I think this is to the last end of the slides. Okay.
Yes.
So maybe I'll look at and go to the next slide, which is basically the conclusion.
Okay.
So what is needed? We need neutralizing antibodies. All right. We want to reduce to prevent infection. We want something that is easy to detect. We want to make it easier for the vaccine developers to generate the alternative, all right? But we must not forget the T cells. It's easier to measure antibodies, of course, but I do hope the vaccine developers will look at what kind of T cells we have. However, we do know from the two of the studies or three that I presented that there are non-neutralizing coronaviruses, which at a high level in patients with severe disease. And they are absolutely present in the presence of the virus itself. Therefore, if we use antibody testing to drive policy, until researchers know what percentage of survivors are producing these antibodies, we need to consider carefully, especially when we are going to evaluate the efficacy of these vaccines. Maybe the talk has been a little bit up, a little bit the high side, but I hope you got a little bit of what I was able to tell you. Okay? And if you need to contact me, you can contact me at this email at the bottom. Thank you.
Thank you very much,
Yeah.
Professor. Yeah, it's really informative, actually. Yes. So, and we already have quite questions here. Like we have more than 4, I think. So we will continue in discussion, and I will start from the first question. It's from Fadlina Tin Naomi. She has 2 questions, and the first one is, what makes D614G mutation is more virulent than the wild type one? And then the second one, could you treat SARS-CoV-2 disease by using the treatment that has been applied previously to SARS-CoV? Yes, that's the 2 questions.
All right. With regard to the current mutation that is circulating, all right, some people call it the super spreader, but really a super spreader would be a viral infection that basically is able to replicate much faster than the first time around. Okay, as we expose ourselves to the virus adapts to our body, so the replication will be faster. And that's when it gets on the term. Of course, you need a number of mutations in these viruses, but in this group of coronaviruses, it's not that easy to cause mutations that will make a change in the lineage. So the lineage is going to be the same, all right? But does it give you more severe infections? No, I think it's the same, except that maybe it's causing more infections because instead of one person infecting one person can infect 5 people. So your R factor increases. So that's the first one. The second one is with regard to treatment, right? Now,
Yes.
I think with this is like under the whole group of coronavirus, there can be common modes of treatment that can be used, all right? But it depends on the population. It depends on the immune status of the individual. As you can see, this disease is attacking the immune system. Basically, it's dampening all the, it's infecting cells, part of the immune system as well. So therefore, the background of the patient's own immune system will also play a role. So it has to be on a case-to-case basis. I mean, they've been trying so much, as we have seen in the literature, but they haven't found the perfect one yet. Okay, so it's like a combination of things. You know, in a different population it works, in another population the other works, and so on. Yeah.
Yes, so it works differently actually,
Yes.
right? Yes,
Right.
that's right. And also we have another question from Firdaus Syed. How to make our immune system strong naturally to fight any kind of virus?
Okay. This question, I would say I'll answer from 2 perspectives. One from the scientist and one from the normal human being, okay, that I am. Okay. Of course, from the scientist perspective, when you want to make your immune system stronger, is to stimulate it with small doses at a time. That's why in Asia we say we are from young age we are sensitized to a lot of things because we play in the mud, we play in the river, you play everywhere. So you're sensitized. In that way you're giving it small doses and the immune system has seen it, so the next time it's better. Right? Yeah. So we can sensitize it that way, and that you can then make a stronger immune system, no doubt. Naturally, yes. From the spiritual side of things, I tell myself this: it's all in the head. If you, I mean, you know what your body produces actually is how your state of mind is. When your state of mind is good, you are performing actually at a near perfect level. But when your state of mind is not so good, then maybe the wrong factors are being released, or it's not as good as it is. So that's the spiritual side of it. But there are many things out in the market that people are selling, which you can also use. But you must believe in them, lah. So if you believe in something works, it will work. But if you are skeptic, it won't work, right? Yeah.
Okay, so people now should let go of their children playing around the playground.
Yeah, I think so.
Yes.
Yes.
Okay, and then another question is from Bintang Anugrah. As far as I'm concerned, SARS-CoV-2 is a virus that can easily mutate it compared to the other viruses. So the vaccine development should be very challenging. Is there any effective way to design a vaccine that can be efficient to any type of SARS-CoV-2? And should we apply the bioinformatic approach as well?
Okay, actually the first part I don't quite agree. I don't think it is the virus that can mutate so easily. All right, it'll take quite a bit of time before it can really cause a big change and to change the lineage per se. All right. Well, where the vaccine manufacturers are concerned, they have to be on their toes. Okay, if you come up with one part of the spike, you're not using the whole spike. If you use part of the spike, and in times there's the changes in the spike and your vaccine doesn't work. When you give the whole spike, there are so many epitopes your body can respond to. But when you give a subunit of the spike, then it's only that part of the antibody of the spike that you get a response to. So it depends on the type of vaccine. All right. So if there is a change, and fortunately or unfortunately, that particular part has changed, then the vaccine that you had may not work. Then vaccine manufacturers have to redo that part and maybe add on another part of the spike. Yes, it'll have to change. Yeah, so it'll be a challenge for them. You can use the bioinformatic too, no doubt. You can use that, but you have got so many epitopes, and right now you don't know which area is the most immunogenic. If you do know, then that is the area you would use. Yeah.
Yeah, okay, yes. So that's pretty clear actually. Yeah, so we reach the end of this session, Professor Shamala, and yeah, I would like to summarize this session from you. Yeah, so to address this COVID-19 response in our body, we need to understand the interaction between virus and the host immune response and the pathogenesis of infection to identify the valid therapeutic tools. Our immunity does not rely entirely on antibodies, and then the serological response differs among patients based on the severity of the patient itself. Okay, so thank you very much for the very interesting and clear presentation.
Thank you. Most welcome.
Yeah, I believe this session give a lot of insight and knowledge more about the situation that we have now, that we are hoping that we can get the condition better a lot. Yeah, so we talked previously about the research. We cannot do the research like the normal situation because of this, and then so yeah, again we should wait for the better situation for everything. And I hope
Rika, I just have one little thing, right?
Yes.
I welcome any collaboration,
Sure.
but to note not necessarily in COVID-19 because my passion is also in dengue, and anybody there wants to work on dengue, and you can look at my papers, and we can see what we can do together. Why not?
Yeah, sure. Of course, Professor.
Thank you, Professor.
Yes, Paromat, yes. Okay, thank you so much. Again, thank you so much, Dr. Shamala, and hope to see you. We're coming to Malaysia, or you're coming to Indonesia. Yes, maybe next year or in the future. Yeah. So, and thank you very much for everyone. So I conclude this webinar, and thank you for all the attendance. And we hope you have learned and enjoyed these two presentations today. Thank you and good afternoon. And I'll hand it back to the Master of Ceremony. Thank you so much.
Thank you, Dr. Rika. Ladies and gentlemen, we have now come to the end of the 16th MIPATalk series, which probably means please forgive us for our shortcomings. I'll see you again next time, perhaps in the next event. May all of you always safe and healthy. Allow me as a Master of Ceremony apologize for any mistakes. Have a nice Thursday. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you so much.